Okay, let us get started for today. I hope the PPT is visible. The PPT is visible, right? Mhm. Okay, so welcome everyone to the week 11 for the molecular biology PMRF-NPTEL session. This week we are going to deal with different modes of genetic engineering. We are going to learn how we have developed different tools for in manipulating the genome of different organisms and learn how to use them as well. So, a quick introduction for myself. I am Anuvab Chatterjee. I'm a PMRF fellow of IIT Kanpur, and I will be taking this PMRF NPTEL session. As you would know, that these sessions are designed so that we can discuss different problems from the previous year's run of the course, so that we are better able to solve this year's quizzes and also get a better understanding of the concepts behind the quizzes as well as the course. So, you would know how we do this is that I will show you a question from the previous year's run of the course and ask you to try and solve it. And then we will try and solve it together so that we get a better understanding of how exactly to tackle such problems and get overall get better knowledge of the entire thing. So, does anyone have any questions from this week's videos on genetic engineering specifically? Okay, I guess not. So let us get started for this week's course. And this is the first problem that we'll deal with. The question is, in the RecF pathway of homologous recombination, bacteria use a repair mechanism specifically for blank in DNA. What kind of repair does RecF do? The options are double-stranded breaks, single-stranded gaps, mismatched bases, and DNA crosslinks. So you can put in what you think is the correct option in the chat. Rupsha and Oropi both say it is B, single-stranded gaps. Srijani also agrees that it is single-stranded gaps. So let us look at it. So why do we go into this? Why do we care about genetic repair as well as genetic engineering? So there are different lots of genetic disorders that appear in us, in humans, which occurs due to double-stranded breaks or single-stranded breaks or overall problems with the genome. So there are some kind of mismatches, there are frame shift mutations, which make the genetic information present in our DNA not the correct one. For example, in case of Huntington's or leukemia, there are different mutations or additions or insertions in the genomic DNA that causes specific genes to start malfunctioning. And once that occurs, then this kind of diseases just keep on accumulating and cause a lot of problems. You probably would have heard of more famous ones such as Alzheimer's or Parkinson's. Cystic fibrosis is also a good example, muscular dystrophy. And these all occur when the body is not able to by itself repair the changes that have occurred in the DNA. And thus, it is a very important field of research in which we are trying to figure out better and better tools to actually edit artificially the DNA present in our cells so that you can make the problems go away. You can fix the disease. So, for example, there are different, these are a example of different disorders that happen, and then the techniques that we use in order to fix them, such as HIV-1 infection. You use a adenovirus mediated or a mRNA mediated targeting strategy to change the different DNA sequences associated. In case of leukemia, do the same. Thalassemia, which is in disorder in which BCL11A gene gets mutated, you again use a mRNA-based targeting strategy to change the DNA. Sickle cell anemia, again, BCL11A gets disrupted, which causes the red blood cells to take a sickle shape and reduce the ability to transfer oxygen to different parts of the body, especially in capillaries. And again, target strategy includes aiming for the specific cells in which this mutation has occurred, or in cells which this is the most, the most of the problems are occurring. Or in the case of Parkinson's disease, where a lot of different genes could be affected, and depending on which gene is affected in which individual, you target that particular gene using different methods, and these are delivery vehicles. So this is where how you put in the mechanisms to edit the genome inside. So these can include adenoviruses or adeno-associated viruses, or just plain mRNA, which can go in and edit the genome. So, as I said, there has been a lot of research in this, and this is the strategy that we generally use for treating genetic disorders. So, what we use is known as a ex vivo approach, or it could be an in vivo approach. So, for an ex vivo approach, for example, in case of a disorder in T cells. You can collect some of the T cells from the patient, you culture them in a media and a dish outside the patient, and then you edit the cells out, the genome of these cells outside the patient using different tools which we'll learn today, such as zinc finger nucleases, TALENS, or CRISPR. And then you take these transformed cells and put them back into the patient where they actually help ameliorating the disease. The other way you can do it, where you can't take out the cells, such as in case of nervous disorders, such as Alzheimer's, Parkinson's, or spinal muscular atrophy. You actually target the cells in vivo. So you want to target the neurons themselves. So you put in adeno-associated viruses or mRNA directly that will approach these cells and do the genomic editing in the cells of the body. That is called an in vivo approach. And when you take the cells out and edit them outside the body, it is known as an ex vivo approach. So this is how we treat genetic disorders, and now we are going to learn more about how exactly the cells or the bacteria by themselves try and do this. So they do this by a process of recombination. We'll learn what this is, but it is essentially a process in which you change the DNA or modify the DNA. And this can occur in two different ways. One is homologous recombination, and another is non-homologous recombination. These are the native ways in which cells deal with DNA damage. In case of homologous recombination, they use a template which can be used to fix whatever DNA damage there is. Say you have a single stranded break. So wherever you had a DNA, one of the strands actually has some bases missing. So you can use the other DNA strand to repair it, or you can use the corresponding strand on the second chromosome. So all of us have a pair of chromosomes for all of the DNA that we have. So even if there is a damage in one of the single strands of one of the chromosomes, the other is absolutely fine. So we can look at the DNA sequence of the other chromosome and try and fix the original one. So when you use a template to fix DNA damage, it is known as homologous recombination, and it could be bacterial or eukaryotic. These are very similar in nature. We will see how that works. Or there is non-homologous recombination where you do not use a template. You do it some other way that we will learn. This can include conserved single-stranded repair, non-homologous end joining, transposition, or VDJ recombinations, and we'll see all of this in a bit. So, what is homologous recombination? So, as I said, all of us have two copies of each chromosomes, and this helps out a lot if one of the single strands of one of the chromosome is broken. You can just look at the corresponding second chromosome and fix it just by looking at the other chromosome, and that is why having two chromosomes helps us a lot. So, homologous recombination is a genetic recombination process characterized by the exchange of genetic information between two closely related or identical molecules of nucleic acids. So, closely related or identical molecules, the other chromosome essentially is what helps out the most. These molecules can either be double stranded or single stranded and are composed of DNA in cellular organisms. Although in viruses, RNA may also be involved in the recombination process. Mostly it is involved in repair mechanism and in meiosis of prokaryotes and eukaryotes. So you would have also heard of recombination during meiosis if you have come across it. So you know there is something called crossing over that happens, which gives rise to a lot of uh, adaptation due to genetic shuffling. In that, what happens is that two identical chromosomes come together during meiosis and they form a chiasmata in which part of the chromosomes actually overlap and are exchanged between the two chromosomes. This causes a lot of changes of information of the DNA present in the crossing over region, and this introduces the variability that is required for evolution to work on. So this is a process that occurs normally during meiosis, but also during DNA repair mechanisms. This kind of similar thing occurs, but not on the chromosomal scale, but only on the DNA single-stranded or double-stranded scale. So this is a huge recombination, but this big of recombination does not occur during repair. So homologous recombination, there are uh, different ways prokaryotes do it and eukaryotes do it, but they are similar in some sense. The two major ones that we will deal with today are the prokaryotic ones, the RecBCD pathway or the RecF pathway. The BCD pathway is used for double-stranded break repair, where both the DNA strands are broken apart, and this can occur due to many different reasons. It can occur normally due to stress in the cell, such as oxidative stress, or it can also occur due to extra environmental factors, such as radiation. As we would have heard, UV radiation causes cancer because it causes uh, different problems with DNA replication, such as thymine dimers get formed that get skipped during DNA replication because DNA replication machinery does not understand what the thymine dimers are, and that causes lesions in the DNA where there is a gap where the thymine dimers or the bases which are damaged due to radiation are, and in that cases or in case of even stronger radiation, both the strands of the DNA could break, and in that case RecBCD pathway comes into play. And this repair can also be start initiated due to chemicals that the cells are exposed to. There are many mutagens that are present, such as ethylene bromide, and etc., that causes mutations in the DNA as well as lesions. And these most of the time act on one of the single strands of the double-stranded DNA. And in that case, RecF pathway takes over. So we'll discuss RecF pathway in some detail. So what is the RecF pathway? RecF pathway is a homologous recombination pathway. That is, it uses a template to actually fix the DNA, and this is majorly seen in prokaryotes, especially bacteria, in which the repair mechanism for single-stranded DNA, it is a repair mechanism for single-stranded gaps in the DNA. So when this also can come up and fix double-stranded breaks if the RecBCD pathway is somehow non-functional. But majorly it is used for single-stranded gaps. So how does it work? First, so there are a lot of different enzymes that are involved. The first is RecQ, which is a helicase that unwinds DNA. You know that with the DNA normally is in a supercoiled state. So first you need someone to unwind it from the double supercoiled state, and then also pull them apart in from the double-stranded helix that they have formed with the hydrogen bonds. Then RecJ nuclease degrades the strand with the 5' end, leaving the strand with the 3' end intact. So once the damage has been done, the gap actually needs to be extended a bit. So even if there is a one nucleotide gap, you extend it a bit so that the template, which will come from the other chromosome, can invade and then can be used to repair. So you degrade part of the damaged single strand. Then there is this protein RecA that binds to the strand at 3' end, and RecF, OF, and R aid and stabilize RecA in this process. RecA then goes and finds homologous DNA that can be used to as a template, and then it exchanges places with identical strand in the homologous DNA through strand invasion. We'll see in a diagram and will be more clear. There is a holiday junction form that also we will see, and the holiday junction. So once the holiday junction is formed, essentially the template sits on top of the broken DNA where there is a gap, and DNA replication enzymes will come in and just use the template to repair the DNA. So despite the differences in proteins and specific mechanisms, the RecVCD and RecF pathway actually are pretty similar. They both require a single-stranded DNA from the 3' end and the RecA protein for strand invasion. Additionally, the pathway exhibits similarities with the phases of branch migration and resolution of holiday junction. So we will go through a diagram so that it is a bit more clear. So say you have eukaryotic double-stranded DNA, and the pathway is named differently in bacteria and eukaryotes, but overall it functions very similarly. These are the partner enzymes in bacteria and eukaryotes. So say you have some kind of radiation or mutation or chemical agent that causes break in the DNA. So this part of the DNA is disrupted. Once it's disrupted, so one part of the DNA is now hanging without no partner because there is no one to hydrogen bond to. So the first in the protein that comes here is called the SSB protein, the single-stranded binding protein. They come and bind to the single strand that is now exposed because the other strand is gone. And these ones stabilize the single strand that has no damage. Similarly, there is red J. This enzyme comes in, detects that there is a damage, and then starts cleaving the DNA from the 3' to 5' direction. So it starts removing some of the DNA off. So it removes more than that was damaged so that the other proteins have space to come in. Then there is this REC F, R, and O complex that forms and removes the single-stranded binding proteins and allow REC A. REC A is the actual uh, what do you call it? Actual protein that causes the repair to sit on the single strand, and once it sits, then RecA goes and looks for DNA that is identical to this single strand, which will be complementary to this broken strand. And it looks for in the other chromosome. And once it finds the other chromosome, so this is the other chromosome in light pink, it forms something known as a holiday junction. The holiday junction is this cross-like junction that you can see right here. So one of the strands from the other chromosome actually comes, invades this double strand, and then goes over it. So that the gap that was present is actually covered by the other chromosome. And then finally, RecQ, as well as which is a DNA polymerase, uses this strand as the template and reforms the broken strand. And once the broken strand is completely formed and it joins to the other end that was here, the holiday junction is unwound, and then sometimes it can happen that there is crossing over where the holiday junction breaks right here in these places. So part of this chromosome goes here, part of this chromosome comes to this place, or it could be non-crossing over where it is just unwound and the chromosomes go back to their original places. But essentially, what you have to keep in mind is that there is a holiday junction that forms, and the other chromosome acts as a template in this and helps in the repair of the break that occurred, the single-stranded gap that occurred in the first broken strand. And that is why it is called homology repair, because you are using a template to repair the damage. So in RecF pathway of homologous recombination, bacteria use a repair mechanism specifically for single-stranded gaps. Any questions? Okay, it seems it is clear. Moving on. In tyrosine recombinase, one strand in each site is cleaved, leading to the formation of a blank. The options are a double-stranded break, a holiday junction, a single-stranded gap, or a fold structure. You can put what you think is the correct option in the chat. So tyrosine recombinase is part of non-homologous repair, and we'll look into that as well. But what does this uh, during repair? It does something called, so it cleaves one of the other strands as well and causing formation of what of the following: double-stranded break, holiday junction, single-stranded gap, or fold structure. Research is coming in. Orupi and Chanda both say it is B, holiday junction. Anyone else? Rupsha also says it is B, holiday junction. Let us look at it. So now we are going to talk about site-specific recombination, which is a different form of repair. So this is also called conservative site-specific recombination, in which a defined sequence between the two DNA is used and recombined between the two strands. This process is carried out by proteins called recombinases. So recombinases are proteins that actually cut and paste DNA, but depending on what their use is, they can actually they are majorly used in repair, which brings together specific ends of DNA, forming a synaptic complex resolving one of the three outcomes, which could be insertion, inversion, or deletion of the site. We'll see how that works. So, based on amino acid homology, they could either be serine recombinase that we are talking about in site-specific recombination, or a tyrosine recombinase. So, a serine recombinase cleaves all four strands that are involved in the exchange. But in tyrosine recombinase, one strand in each site is cleaved, leading to the formation of a holiday junction. We'll see that how that works. But the most famous example of tyrosine recombinase is the Cre recombinase that recognizes the LOXP site. If you have a bit of idea of how recombination and genetic engineering in mouse work, Cre recombination is the most used technique in which Cre recombinase recognizes a site called a LOXP site, and anything between two similarly directed LOXP sites can be cleaved out by Cree. So that is used a lot in the genetic recombination in mice, where you want to study the loss of function, say, of a particular gene. You put LOXP sites at ends of the gene, and Cree will just cleave it out in vivo, so your mouse does not have that particular gene anymore, and you can study different things in the mice. These could also include lambda integrase or ZER-CD resolvase complex, or on the other hand, serine recombinase, which cuts all four strands, include the HIN invertase or the TN3 gamma delta resolvase. So let us look at how mechanism of tyrosine recombination works. So, I would ask you to pay some attention to this. So this is the gap that has been formed, the one site that has been damaged due to some radiation or chemical agent. So the recombinase will first cut one strand on each site. So it will cut one of the strands here and a similar strand on the other chromosome. The cut strand is now exchanged between the molecules. So this goes here, and this goes here. So you see there is a crossing over and this X type holiday junction formed again. And for this is the formation of a holiday junction. And then there is a cut at the second site, which is right here, which is in the other strand, which was perfectly fine. This strand is broken, and then strand exchange will occur. So you see, this is black, then yellow, black, then yellow. So essentially, you exchange partner strands, and then the holiday junction is resolved by cutting and pasting the DNA. So essentially, in this kind of recombination, this particular wherever the damage is, that can be resolved, or if there is no damage, also if the tyrosine recombinase in case of 3-lox-P knows that this is a lox-P site, then this particular recombination will occur in which similar sequences are exchanged between the two chromosomes. Similar technique is used for cross 3-lox-P deletion as well. If the same sequence is present on the same strand, then this strand essentially gets circularized and cut out, and these two ends joined, so you do not have anything here. So this is how a tyrosine recombinase forms. It goes via the holiday junction formation and strand exchange between the two chromosomes. Why is CSSR important or conserved site-specific recombination? It allows for DNA repair. It allows for precise cut and replacement of damaged DNA strand, maintaining genomic integrity of E. coli. It allows for gene regulation. For example, the lambda phage uses CSSR mechanism to switch between lytic and lysogenic cycle. So once you cut and paste the strands on other ends, it can also give rise to different proteins being activated or inactivated because obviously you're changing the genomic sequence. Genomic rearrangement, PSSR causes gene rearrangement leading to various combinations such as inversion, insertion, or deletion of gene segments. For instance, switch in flagellin component of flagella in Salmonella is mediated through CSSR. Horizontal gene transfer bacteria often use this mechanism to achieve new traits from other bacteria or mobile genetic element leading to bacterial adaptation and evolution. Or it can also help in development. Many organisms use site-specific recombination methods to regulate tissue-specific gene expression and cell differentiation during development. And the other methods include use of this CSSR for developmental biology as well as genetic engineering. So tracking of cell lineages during development of Drosophila was done using the flip FRT system, which is also a form of site-specific recombination. So flip FRT system works very similarly to Crelox-P, in which FLP and FRT are the protein and the sites that it recognizes, and it does recombination depending on that. And when the recombination occurs, you can have a particular protein such as GHP being activated, and you can trace where whichever cells there is recombination and track them. And CSSR are used to manipulate and engineer genomes of organisms like ablation of genes, so using Crelox-P, or using Crelox-P again, you use a different form of Cre, which is Cre-ERP2, which is Cre fused to estrogen, modified estrogen receptor that can be activated with a synthetic ligand called tamoxifen, and you can choose when the recombination happens. And whenever the recombination happens at that stage, then the mouse or whatever organism you're dealing with no longer has the gene that you're interested in, and thus you can uh, investigate time specific time specific loss of function of that particular gene. So induction of gene expression at specific time during development can also be done using CSSR. So in tyrosine recombinase, one strand is in each site is cleaved, leading to the formation of a holiday junction. Okay, next. The recombination process occurs between specific sites on the VDJ segments in genes for generation of. So VDJ recombination happens majorly in what? Immunoglobulin and T cell receptors, antibody and cytokines, ribosomal RNA and proteins, enzymes and hormones. You can put in whatever you think is the correct option in the chat. So VDG recombination occurs in which of the following things? Hemoglobin and T cell receptors, antibodies and cytokines, ribosomal RNAs and proteins, enzymes and hormones. You can put in what you think is the correct option in the chat. Okay, so I think no one has an idea of this, so we'll go and look at it. So VDJ recombination. So VDJ recombination is a big thing in antibodies. So an immunology in which VDJ recombination is used to produce this huge uh, VDG recombination is used to produce a huge variety of antibodies that can detect antigens that are thrown at our body. So, if you have ever thought of how our body can detect various different external antigens, so various different bacteria or viruses that attack us every day, and then mount an immune response, so they do it via VDG recombination. So it is a specialized set of DNA recombination mechanisms that impart enormous diversity to B cell and T cell receptors. So there are like a billion variety of bacteria and viruses out there. So how does the body recognize every one of them? So that is this is because we can produce a similarly huge variety of antibodies that are produced via the VDG recombination process that we will see. So this recombination process occurs between specific sites on V, D, and J segments in genes for the generation of BCRs and TCRs. So B cell recognition elements and T cell recognition elements. And these recombination are called sites are called recombination signal sequences. This can be either 12 base pair or 23 base pair long, and we'll see how they work. So for B cells are the ones that make our antibodies. So they have three different regions in every one of the antibody producing genes. One is a V region, one is a D region, and one is a J region. And then there is a constant region which remains constant and does not take part in recombination. Recombination. So the V region has 40 different DNA segments. The D region has 23 different DNA segments, and the J region has 6 different DNA regions. During recombination, and these occur spontaneously in B cells, one of the V regions, D regions, and J regions, along with the constant region, come together. And once they come together, when they are transcribed into the RNA, so 1 out of 40, 1 out of 23, 1 out of 6, so these V, D, and J's are chosen to form the final antibody. So what happens essentially is that you have 40 into 23 into 6, that is the amount of diversity that can occur in the one gene of the antibody. So again, the antibody is made out of two different what do you call it? Two different chains. One is called the heavy chain, and one is called the light chain. So in heavy chain, this is the amount of changes that can modifications that can occur. Similarly, in the light chain, the V region can go from 30 to 38, and J region from 1 to 5. Again, there is 38 cross 5. That is the amount of combinations that can occur, and then you have a final antibody that has both the light chain and the heavy chain. So essentially the number of combinations you can have is 38 into 5 into 40 into 23 into 6. Very huge number from one antibody gene. And this is how B cells produce this many different kinds of antibodies, because they have the ability to make different kinds of antibodies preset in their genome. because of this VDJ recombination. Similarly, in case of T cells, so T cells are the cells that can recognize antigens directly. They have receptors that are present on the cell surfaces. They also have V region, B region, and J regions, which come together after rearrangement to form a specific combination. And after transcription, again, you have a huge variety of V, D, and J combinations that can take place, as the T cells can recognize a wide variety of antigens. So the recombination process occurs between D and J segments in the gene for the generation of immunoglobulins and T cell receptors. Any questions? Please put in the outside. Any questions anyone has? Okay, next question. NHEJ, non-homologous end joining, is a mechanism for repair of blank in DNA. Single-stranded breaks, double-stranded breaks, mismatch bases, or cross-linked DNA. So what is NHEJ a mechanism for? You can put in what you think is the correct option in the chat. Mhm. No. Mhm. No. Yeah, you can put in what you think is the correct option in the chat. NHEJ is a mechanism for repairing of DNA in which of the following uh, cases? Is it single-stranded breaks, double-stranded breaks, mismatch bases, or cross-linked DNA? Okay, we have answers coming in. Ruksha, Srijoni, Rupi, Koel, and Chanda, all of them say it is B. Double-stranded breaks is what NHEJ repairs. So let us look at it. So we were talking about homologous repair, and the other is non-homologous repair, and part of non-homologous repair is called non-homologous end joining. So non-homologous end joining, so homologous repair, you saw that there is strand invasion from the other chromosome, and looking at the corresponding strand that is fine in the other chromosome, the DNA repair mechanisms actually remake DNA. But in non-homologous end joining, nothing like that happens. It is that there is a break in double-stranded DNA. You just take the two pieces and join them together. Just cut and paste. There is no repair per se. It just it is a repair. It joins two strands together. But if there are any bases missing, then there is no addition of bases. It just joins the two ends together. So non-homologous end repair, end joining, is a mechanism for repairing double-stranded breaks in DNA. Unlike homology-directed repair, which requires a homologous template, which is normally the other chromosome, NHEJ directly ligates the broken ends without the need for a template. This makes NHEJ an efficient process that can operate in both dividing and non-dividing cells, and the term homologous end joining was coined by these two people in 1996. So NHEJ is often guided by short homologous DNA sequences called microhomologies, but this is not often required. These sequences are typically found in single-stranded overhangs at the end of the double-stranded breaks. When the overhangs are compatible, NHEJ can accurately repair breaks. However, if overhangs are not compatible, imprecise repair occurs, leading to loss of nucleotides. So what happens is that if there is a double-stranded break, but the double-stranded break is sticky-ended, which means that there are overhangs on both the ends, something that occurs often that we have seen using restriction enzymes. Then during non-homologous end joining, the systems, the different proteins, they will do is chew the DNA back so that the overhang is completely removed and you have blunt-ended DNA that are joined together. So any information that is present in these overhangs is lost. So inaccurate NHEJ repair can result in loss of genetic material and lead to translocation and telomere fusion. These events are hallmarks of tumor cells, highlighting the importance of proper NHEJ in maintaining genomic stability. It is a widespread mechanism in all biological systems. In mammalian cells, it is three dominant pathways for double-stranded break repair. However, in yeast, homologous recombination tends to dominate. When NHEJ is inactive, double-stranded breaks may be repaired by alternate, even more error-prone pathway known as microhomology-mediated end joining. This is even worse than non-homologous end joining. So in MMEJ, resection reveals short microhomologies on either side of the break, guiding the repair, and this causes a lot of deletion of DNA. So you'll see how that works in a bit. But first, let us deal with non-homologous end joining. So in case of non-homologous end joining, you have double-stranded breaks on two ends. First, the heterodimer Ku7080 comes and stabilizes these two ends. This recruits DNA PKCs and then nucleases such as Artemis, and they trim the DNA till you have two compatible ends. The compatible ends could be blunt ends, and then there is a ligase complex that comes in and essentially joins the two ends together to form a repaired DNA break. So it is a pretty fast process, and if there is only a clean break between the two strands of the DNA, this is the best process that you can use. But if it is a big break in which a part of the DNA is chewed up, which is causes an overhang to form, in that case, this will cause deletion of different the genetic information because all this overhang will be chewed off by the DNA nucleases, and then the two ends will be joined together. So what are the use of different enzymes? Ku750 and 80 bind to DNA ends and recruits other NHEJ proteins. DNA PKCs phosphorylates and activates the nucleus Artemis and helps removing the broken DNA ends. Artemis is the nucleus process the DNA ends, removes flaps and loops. DNA poly gamma and delta end filling of the processed ends. Ligase 4 ligase that joins together the two ends and the other cofactors that helps in stimulating the ligase activity. So NHEJ is a mechanism for repairing double-stranded breaks in DNA. Next, transposons are segments of DNA that can move from one locus to a relatively non-specific site in the genome are also known as jumping genes, intronic sequences, exons, or regulatory elements. So transposons are also known as what of the following? A lot of answers coming in. Chanda Devarkar says it is A, jumping genes. Srijoni, Koel, and Orupi also agreed that it is jumping genes. Ruksha also says transposons are known as jumping genes. So let us look at it. So what are transposons? These are called jumping genes. These are segments of DNA that can by themselves move from one region of the genome to another region. So transposons are segments of DNA that move from one locus to a relatively non-specific locus in the genome. And so these are essentially just normal genes that are present in the DNA that one day will decide that I don't like the environment here. They just up and then go to some other place and join in together in that other DNA segment. So they can non-specifically jump from one place to the other, and that they can do it any number of times they please. So movement of transposons can occur with the duplication of the element. So they are two classes of transposons. One is called the copy and paste, another is called cut and paste. Copy and paste transposons actually do not leave their original site. but just copy themselves into another site in the DNA, whereas cut and paste is what is shown here, where the transposon will just remove itself and then join it somewhere else on the target DNA. DNA transposase carry inverted repeats at the ends as recombination sites, flanking a recombinase proteins called transposase, which brings about the recombination. And retrotransposons, these are the copy and paste ones, contain LTR and genes integrase and reverse transcriptase activity. So interestingly, transposons are have all the information they need to actually do this transposition inside themselves. So they majorly need a recombinase that will do this cutting and pasting, and that is present in the transposon itself. So it is a self-sufficient jumping gene that contains everything required. So it also has something called homologous ends. So these are random short sequences that can help in homologous. We have seen how homologous end joining works, where if there is some part of the DNA which is identical, that the DNA can essentially push itself together into a new position using a recombinase. So a transposable element or a transposon actually has the transposase, which is the recombinase, as well as these homologous ends that it uses to just jump from one place to the other. So as I said, there are two different types of transposons. First is copy and paste, the other is cut and paste. So cut and paste is easy. We'll discuss this first. So cut and paste transposons have the transposer enzyme inside it. The transposer enzyme cuts these two ends, excises the DNA, and then it goes and helps in integration somewhere else as well. So essentially, it cut and paste ones, they move from one place to the other. Copy and paste, they do not move from one place to the other, but they make a mRNA first. So they transcribe a mRNA, and also they have the mRNA forms a protein which is reverse transcriptase. So the mRNA by itself can be reverse transcribed into a complementary double-stranded DNA. This double-stranded DNA then also uses the retrotransposons LTRs to integrate. So it has everything required to form another copy of itself and then integrate somewhere else in the target DNA. And then there are non-LTR transposons which don't have enhancer sequences on both ends, but they also can transcribe into an mRNA and target primes reverse transcription. So they go near a target first and then do the reverse transcription to integrate. So these are very interesting genes, transposons, that can just go from one place to the other, and they have all the essential utilities that is required for them to go from one place to the other. So what does such kind of transposons and transposition does to the genomic DNA? The first thing that you can guess, it alters gene expression. So if the transposition occurs inside a gene that was important, then it will just go in transpose itself inside the gene and disrupt the gene's function. So it can cause different gene expression changes. The IS element, which is present in the transposons, can cause mutations, REC-dependent recombination, or site-specific recombination between the transposable elements in the same molecules can cause rearrangements such as deletion or inversion. IS1 element can cause deletions. TN3 and gamma delta resolvase can cause resolvase reaction through deletion and mutation in general. So transposons cause a lot of problems essentially. They cause a lot of changes in gene expression, but this can actually also give rise to new functions of genes because of this transposition occurring, such as in case of maize. Maize is the uh, maize is what is you would also known as corn, the one that makes popcorn. So that maize actually has a transposable element that causes different kernels to have different colors. And there is a lot of research done in early 1970s, and that is where it was discovered. So they discovered that different kernels have different colors, but they could never find the reason for it, and that is how transposons were discovered. So transposons are segments of the DNA that can move from one locus to a relatively non-specific site in the genome known as jumping genes. Next, in MMEJ, which is microhomology mediated end repair, end joining, end resection reveals short microhomologies on either side of the break, guiding the repair process, DNA replication, transcription, or translation. You can put in what you think is the correct option in the chat. Okay, we have a number of options that have come in. So Oropi Ruksha Chanda also A, repair process. Devadita Srijoni Koel also say A, repair process. The MMEJ intersection reveals short microhomologies on either side of the break, guiding the repair process. So you're all correct. Mm, so you are all correct. So MMEJ is microhomology mediate end repair. So when non-homologous end joining is not working, then this is what takes over. So in case of microhomology end joining, what happens is that instead of just cutting and pasting, like in case of non-homologous end joining, the DNA repair mechanism involved actually starts chewing the DNA till it finds something called a microhomology between the two strands. So these are specific sequences that are not very common. So it will start chewing and doing a lot of damage to the DNA till it can reach the microhomology region present on both the strands and then join both the strands together. So you lose a lot of genetic information till this repair can find the particular microhomology. So this is like the worst kind of repair that can occur. But this is in MMEJ intersection reveals short microhomologies on either side of the break, guiding the repair process. The folding of the zinc finger relies on tetrahedral coordination of zinc ion with two invariant pairs of blank lysines and arginines, serines and threonines, cysteines and histidines, or tyrosines and tryptophans. Okay, so Chanda Srijoni Coel and Ruksha also it is C, cysteines and histidines. Anyone else wants to try? Devadit also it is C, cysteines and histidines. Let us look at it. So now we since we have learned about the different forms of repair and genetic editing strategies that the cell by itself uses to repair or change or modify its DNA, we know that all of these are not very viable for genetic engineering by us. So artificially editing the genome using these techniques can cause lack of precision, low efficiency, are time consuming, limited editing scope, and have off-target effects. So none of the techniques that we have discussed, either being homologous end joining or non-homologous end joining or transposons, are very effective in editing the genome when we say ask it to edit. So you can't specifically edit the genome using the techniques. So, but as we have said, that genetic recombination is a very important tool to have, not only for just fundamental studies of different genomic sequences, but also to treat various diseases. So, in order to do that, we have actually been trying to find different ways of editing the genome from way back when. The first techniques that we had figured out are restriction enzymes. We have seen how they work is that they cut the DNA at the site-specific manner. So these were discovered around 1970s. This was quickly followed by something known as zinc finger nucleases. So the zinc finger nucleases were designed in order to cut the genome at specific regions, and this could be done in a site-specific manner in live cells. So restriction enzymes, obviously, you use them mostly in DNA that you have taken out of the cells, but zinc finger nucleases can be used for live cells. Then we have TALENS around 2011, so a long time later, which were an improvement over zinc finger nucleases, but worked very similarly. And then in 2012, we had the CRISPR-Cas9 system, which was enormously powerful and just left everyone else to the wind. And the people who discovered this actually got the Nobel Prize in 2020 because of the discovery of CRISPR-Cas9. And this is probably the most simple and most effective genome editing system that we have currently, and it is probably something that you would also know about much more than the other techniques. And then we actually upgraded the CRISPR-Cas9 system over time in at doing different mutations to increase the efficiency. And now we are still using CRISPR-Cas9 as our basis for genome editing. So this is actually a very important technique that we will learn about. But first, we'll go through one by one all the different techniques that were discovered beforehand to know how much of a upgrade CRISPR-Cas9 is and how it works. The first one that we're going to deal with is the zinc finger nucleases. But in order to learn what zinc finger nucleases are, we have to first know what zinc finger proteins are. So a zinc finger protein is zinc finger is a compact protein structural motif characterized by binding of one or more zinc molecules, contributing to the stabilization of the folded structure. So these are parts of the protein that can bind to zinc and help in stabilizing the entire protein. The term was initially coiled on the finger-like appearance observed in the presumed structure of transcription factor 3A from the African clawed frog. However, it has been identified in a diverse array of protein structure within the eukaryotic cell. So it is a motif rather than a protein itself. So it is a part of the protein that can fold independently of the other parts of the protein. And for that, it requires zinc divalent cation, and it forms finger-like projections. But we'll see how the protein looks like. The discovery of zinc's presence in Xenopus laevis transcription factor 3A, which is essential for its function, was reported in 1983, marking the first documented instance of zinc requirement for gene regulatory proteins. So, another thing that you have to know is that the zinc fingers, so the finger-like projection of the protein, can actually bind the DNA. So, it can specifically bind different regions of the DNA. So that is why it has some use. So you can imagine it has use in genome editing because it can specifically bind DNA, the protein itself. Subsequently, similar findings were observed with the Kruppel factor in Drosophila. Zinc fingers commonly manifest as metal binding domains within multi-domain proteins. So they are part of proteins essentially. So what does it look like? So they have finger-like projection. This is zinc, and these are the finger-like projection not shown here. Shown somewhere. So this is the motif that you can see. So this is the zinc molecule. We have antiparallel beta sheet at one end and alpha helices on the other end. So this is the alpha helix and these are the antiparallel beta sheets. So 25 of the 30 amino acids in the repeat fold around a zinc ion to form an independent structural domain or module, the finger. So this is called the finger. On the five intervening amino acids to provide the link between the consecutive fingers. Each unit forms a zinc coordinated finger stabilized by conserved amino acids and a potential hydrophobic clustering. The region is rich in basic and polar residues and are implicated in nucleic acid binding. So you can imagine that since the ends of these alpha helices are basic, so they are positively charged and thus can easily bind nucleic acids, so they can bind DNA very easily. So this is how the arrangement looks like. So the folding of the zinc finger relies on the tetrahedral coordination of zinc ion with the two invariant pairs of cysteines and histidine, forming a conserved cysteine-cysteine followed by zinc histidine-histidine pattern. Additionally, each repeat includes tyrosine in the sixth position or phenylalanine at the sixth position, phenylalanine again at the 17th, and the leucine at the 23rd position, a large hydrophobic residue that may interact in creating stabilized hydrophobic cluster in the compact finger module. The 30 amino acid repeat, rich in basic and polar residue, concentrates a significant number between the second histidine and the first histidine, emphasizing its role in nucleic acid binding. So essentially, the zinc finger has these four basic amino acid leading out from it. So this is the structure of the zinc finger nucleus. This is just the zinc finger motif, and it has these basic amino acids pointing out, and these are the ones that can actually bind to nucleic acids. So the folding of the zinc finger relies on tetrahedral coordination of zinc ion with two invariant pairs of cysteines and histidines. The cysteine 2 or the histidine 2 zinc finger motif consists of what? Two alpha helices and one parallel beta sheet, one helix and an antiparallel beta sheet, two antiparallel beta sheets, or an alpha helix and a parallel beta sheet. So in zinc finger, the 16 2 and histidine 2 forms what of the following? You can put in what you think is the correct option in the chat. Okay, we have a number of answers. Chanda and Rupsha say it is B, alpha helix and antiparallel beta sheet. Orupi, Coel and Devadita Srijoni also say it is B, alpha say helix and antiparallel beta sheet. So yes, let us look at it. So this is the zinc finger that we are talking about. We have the zinc molecule in between, and it indeed interacts with invariant two cysteine molecules and two histidines. So as we have seen, it is found in transcription factor 3A, and this new class of proteins was able to bind to specific sequences of DNA. The zinc finger structure is maintained by the zinc ion, which coordinates cysteine and histidine in different combinations. Originally, zinc finger referred to a DNA binding motif in Xenopus laevis, but now it includes various structures coordinating with the zinc ion, and recent classification focuses on fold groups. So common fold groups include the cysteine 2, histidine 2, like classic zinc finger treble clef or zinc ribbon, and cysteine 2 and histidine 2 zinc finger motif consist of an alpha helix and an antiparallel beta sheet, and the zinc ion is coordinated between the two histidines and the two cysteine residues. So you have an alpha helix and an antiparallel beta sheet. So everyone was correct. So now we were talking about the zinc finger motif. Now we are going to talk about the zinc finger nucleases, which are actually used to do the genomic editing. So zinc finger nucleases are synthetic and restriction enzymes created by combining zinc finger DNA binding domain with what of the following? A DNA ligase domain, a RNA polymerase domain, a DNA cleaving domain, or a helicase domain. So how is a zinc finger nuclease made? So you have a zinc finger motif, but what is the other part? So you can put in what you think is the correct option in the chat. So zinc finger nucleases are synthetic restriction enzymes which are what in addition to DNA binding domains. Okay, so Chanda, Rupasha, and Oropi and Cohen all of say it is C, DNA cleaving domain. So let us look at it. So we have talked about this zinc finger motif, and now people figured out that if I have a particular protein motif that can specifically bind to DNA, if I just attach uh, something that can cut DNA, then I have a genomic editing tool. So that is what they did. So zinc finger nucleases are essentially formed of two different parts. One is the DNA binding domain, that is the zinc finger motif, and a DNA cleaving domain that will actually cut the DNA. So zinc finger nucleases are synthetic restriction enzymes created by combining a zinc finger DNA binding domain with a DNA cleaving domain. The engineered zinc finger domains can be customized to recognize specific target DNA sequences, allowing zinc finger nucleases to pinpoint unique sequences within integrate genomes. Leveraging the inherent DNA repair mechanisms in cells, these agents are employed to accurately modify genetic makeup. So it's a very powerful tool to edit the genome. So how does this work? We have a DNA binding domain that we have seen already. So the ZFNs or zinc finger nucleases DNA binding domain typically consists of 3 to zinc finger repeats, recognizes 19 to 18 base pairs. So the zinc finger can part of it can recognize 19 to 18 base pairs. Utilizing three finger array, specific 9 pair target sequence sites can be addressed. Challenges arise from overlapping specificities of individual zinc finger selection methods, including page display and open systems help create tailored zinc finger arrays. Open, developed by the Zinc Finger Consortium, employs a bacterial two-hybrid system for improved customization, offering an alternative to commercially available arrays. So, in order to use the zinc finger nucleus to its best uh, form, you will have to design the zinc finger domains very specifically so that they only bind to the DNA that of interest to you, because these are amino acids and not nucleic acids. So essentially, the amino acids can recognize anything. So they, even though they normally recognize nucleic acids, they, in order to make it very very specific, you have to use three different zinc finger arrays in the same ZFN, and we will see how even two pairs of ZFNs are actually used in order to ensure specificity. So this was about the DNA binding domain, and the DNA cleaving domain is ZFNs use non-specific cleavage domain, generally type 2 enzymes such as FOC1, which require dimerization for DNA cleavage. So standard ZFNs link this domain to zinc finger domains for effective cleavage, and the two ZFNs must bind opposite DNA strands with specific spacing. Protein engineering techniques included directed evolution and structure-based design aim to improve the nucleus domain's activity and specificity of ZFNs. Directed evolution product produced a variant called Sharpie, which, while structure-based design enhances cleavage specificity by modifying the dimensional efficiency. So if we see an image of this, so this should make it a bit more clear. So you have the zinc fingers which are combined to each other. So three zinc finger modules that detect a particular sequence of DNA. And at the end, you have the cleavage domain. Now, the cleavage domain needs to have another cleavage domain bind to it. So essentially dimerization of cleavage domains in order to actually cut the DNA. So what you do is that you design two different zinc fingers, one that binds to the upper strand and one that binds to the lower strand from the opposite end. And when they bind to exact sequences that they were designed for, they have the correct spacing for FOC1 enzyme to dimerize and cleave the DNA in between. So this ensured high specificity of the cut occurring where you require it. But designing the zinc finger was extremely difficult because these sequences need to these proteins. So the because these are amino acids again, they need to be tried and tested again and again to ensure that there is no non-specific interactions which will cause dimerization of FOC1 and cutting of the DNA, but also be lenient enough that actually the dimerization takes place and cuts the DNA. So there was a lot of protein engineering involved for each and individual ZFNs. And the problem was that if you now wanted to target a sequence that you want, then you will have to go through the process again and again to design your own ZFN. So there was a lot of protein engineering involved for this. So zinc finger nucleases are synthetic enzymes created by joining zinc finger DNA binding domain with a DNA cleaving domain. So ZFNs use a non-specific cleavage domain generally derived from type 2 restriction enzymes like FOC1, which requires blank for DNA cleavage. So what does it require? A single-stranded binding domain, dimerization, phosphorylation, or methylation. So for cleavage, what is exactly required in enzymes such as FOC1? Anyone wants to take a guess? So what? So in case of ZFNs, the non-specific cleavage domain, which is made out of enzymes such as FORC1, require what for cleavage? Single-stranded binding, dimerization, phosphorylation or methylation. Okay, so let us look at it. So as I said, fork1 needs to be dimerized. There need to be two different subunits of fork1 to come together to actually cause the cleavage, and this actually lends this lended the specificity for the zinc finger nucleases, because the zinc finger nucleases need to bind to one strand on the top and one strand on the bottom. So two different specific sequences need to be bound by the ZFNs, after which only the dimerization can occur and cleavage. So this improves the efficiency with which the accuracy and specificity of the cleavage by ZFNs. So the engineered zinc finger arrays are commonly combined with DNA cleavage domains such as FOC1 to create zinc finger nucleases. These fusion proteins have proven effective in genome manipulation across a range of organisms such as Drosophila, C. elegans, tobacco, corn, zebrafish, mammalian cells, and rats. Zinc finger nucleases, when applied, induce double-stranded breaks at specific genomic locations, leveraging the error-prone nature of the non-homologous DNA repair pathway to introduce frame-shift mutations into coded sequences of target genes. So essentially, wherever you cut, whenever the DNA will join back together, it will be non-specifically joined, and thus essentially the gene will be inactivated. Additionally, when paired with a homologous DNA donor sequence, so if you want to actually insert something, then you put in a gene of interest that you want to insert, and this will be used for homology directed repair, and that part will be integrated in the genome. Enables precise genome editing through homology directed repair pathway, converting genomic locus to a predetermined sequence. A notable application involves ongoing clinical trial assessing the reception of CCR5 gene in CD4 plus human T cells using ZFNs. This therapeutic approach aims to explore potential ZFNs for treating HIV or AIDS. Overall, zinc finger nucleases exhibited versatility in facilitating targeted genome modifications, showcasing their utility in both research and potential medical interventions. So, FOC1 needs to dimerize in order to cleave the DNA. Next, we're going to talk about the other genome editing methods. The one that we'll talk about is called TALENS. TALENS are created by combining a TAL effector DNA binding domain with a blank, which acts as a nucleus capable of cutting DNA strands. These so TALENS are made of TAL effector DNA binding domain with what? It does it is it a combination of ligase domain, a helicase domain, a DNA cleavage domain, or a polymerase domain? You can put in what you think is the correct option in the chat. Okay, we have answers coming in. Chanda and Nivedita both say it is C, DNA cleavage domain. Let's look at it. So TALENS is the second genetic engineering tool that we are going to study. So TALEN, the word TALEN is actually in two parts. The first is tail, and the last is the N. The tail means transcription activator like effector, and N is a nuclease. So it is a combination of two different things, a tail and a nuclease. So what is a transcription effector like activator like effector nuclease or TALEN? It is a completely engineered restriction enzyme designed to target specific DNA sequences for cleavage. It is created by combining a TAL effector DNA binding domain with a DNA cleavage domain, which cuts the DNA by manipulating transcription activator like effectors tails. Scientists have been able to customize bind to virtually any DNA sequence. When these engineered tails are coupled with nucleus, precise DNA cleavage can be performed. So, what is the advantage of tails? So, tails are transcription activator like effectors. So, these are specific proteins that are released by plants specifically, such as particularly plant pathogenic xanthomonas bacteria utilize tails produced through their type 3 secretion system. And these proteins actually have a lot of different domains, but the most important part that we are interested in is a central repeat domain or a repeat variable domain. And the central repeat domain actually has specific sites which can bind to nucleic acids. So the stranded repeat sequences in between the entire tail, which is this sequence. So there is with the hypervariable 12th and 13th position, known as the repeat variable diresidue or the RVD, the identity of the two residues in consecutive repeats corresponding to sequential DNA bases in tail effectors target sequence. So essentially, all these repeats are there, and this particular two residues can be designed to bind to different nucleic acids. For example, if you have a Ni, it can bind to adenine, Hd can bind to a cytosine, Ng can bind to mm, thymine as well as a 5-methylated cytosine, Nn can bind to R, R is essentially purine, Ns can bind to anything, Nk can bind to guanine, and NH can also bind to guanine. So depending on how you design the central repeating domain of your particular talent, essentially the talent can go and bind to any specific nucleotide. And this was experimentally determined. So now you have actually upgraded yourself from the zinc finger nucleases because we actually now have a map of how to design where your particular talent will go and sit. And the next step was obvious. They just attached a DNA cleavage domain to the end, which is again the FOC1 DNA cleavage restriction enzyme, type 2 restriction enzyme. So this is how talents work. So exactly similar to zinc finger nucleases, but now you have much more control over how to design your talent because you know what binds to what. So you don't have to figure out by trial and error and a lot of protein engineering, like in the zinc finger nucleases, on how which zinc finger will specifically bind to the DNA or sequence of interest to you. You know, whenever you put particular amino acid, it will bind to a particular nucleic acid. So you can design it very easily compared to zinc finger nucleases, and then just join your FOC1 endonuclease to the end, and then you cleave the DNA wherever you want. But the arrangement is identical. One talon will bind to the top strand, one will bind to the bottom strand, and dimerization of FOC1 will cause the cleavage. So it is so what are the applications of Talon? So Talon is a versatile tool for genomic editing. It is instrumental for engineering of iPSCs, so human immuno induced pluripotent stem cells, and it has been used to fix and treat diseases such as sickle cell disease, as well as to combat cancer, and can be combined with other tools such as meganucleus to enhance its capability. In terms of difficulty and cost, Talent falls between the zinc finger nucleases and CRISPR, offering a balance between specificity and engineered simplicity. So Talents, when they came out in 2011, were the major thing, and they were named as the method of the year for how they were much simpler than zinc finger nucleases, and people thought this is it. But then CRISPR came along and then blew everyone out of the water. So talents are created by combining tile effector DNA binding domain with a DNA cleavage domain to act as the nucleus for cutting the DNA strands. Next, now we are going to the final genome editing tool, CRISPR. CRISPR consists of conserved repeated sequences interspersed with coding sequences, electronic sequences, short spacer sequences, or promoter regions. So, what is CRISPR? Actually, what is the full form? Essentially, if you know the full form of CRISPR, you will know this. So, it consists of which of the following: coding sequences, electronic sequences, short spacer sequences, or promoter sequences. You put in whatever you think is the correct option. Okay, so let us go through it. So what is CRISPR? The full form of CRISPR is clustered regulatory interspersed short palindromic repeats. So a mouthful, but essentially, uh, not talking about that. So we are first going to talk about what CRISPR is, and then look at the applications for genomic editing. So CRISPR-Cas9 systems are sophisticated adaptive immune systems of prokaryotes. So these are systems that are used to fight against viral infections and not for genome editing. So CRISPR by itself, so clustered regulatory interspersed short palindromic repeats, is a DNA sequence found in prokaryotes such as bacteria and archaea, originating from previous bacteriophage infections and serves as an adaptive immune system, allowing cells to detect and eliminate similar DNA threats. It is present in about 50% of bacterial genomes and 90% of archaea. And Cas is just CRISPR-associated protein. So CAS, CRISPR-associated protein, is an enzyme that uses CRISPR sequence as a guide to recognize and open up specific strands of DNA and cut the DNA. And the Nobel Prize for Chemistry was won by the Emmanuel Charpentier and Jennifer Doudna for development of CRISPR-Cas9 system. So how did they figure it out? The CRISPR was first characterized by Yoshizumi Ishuna in 1987, but I didn't look at the function specifically. And then people started looking at the function, but actual important breakthrough came in 2012 when CRISPR-Cas9 was projected as the genomic engineering tool that it is now. So we'll see how it works. But first, let us just look at CRISPR. So the clustered regularly interspersed short palindromic repeats. So essentially, in the whenever so bacteria and archaea, which are essentially these small microorganisms, are always at a huge war against viruses. Viruses hunt bacteria, and bacteria try to figure out different ways of defending themselves from the viruses. When a particular virus infects the bacteria that the bacteria has not seen before, then there are different Cas proteins, Cas1 and 2, look for these foreign DNA sequences, and they essentially take part of the DNA sequence and integrate them into the CRISPR repeat spacer array. So if the bacteria survives the infection, next time a similar virus comes in that has the same spacer sequence, the CRISPR-Cas9 system activates. And depending on this sequence, the CRISPR-Cas9 system goes and matches this sequence to any other foreign DNA around in the cell. If it finds any DNA, it stops it up. So essentially, this acts as an adaptive immune system for the bacteria which prevents recurring viral infections. So once the protospacer from the virus has been integrated in the CRISPR array, the bacteria is ready to fight off a new infection because the CRISPR system will recognize this foreign DNA from the virus as the viral DNA and start chopping it up. We'll see how that works, but for now, CRISPR consists of conserved repeated sequences interspersed with short spacer sequences. So in the adaptation stage, the blank complex acquires a protospacer from the invading viral DNA and integrates it as a new spacer into the CRISPR array. So it is Cas9, Cas1 or 2, Cas12a or Cas3. Which of the Cas proteins helps in adaptation of the Cas9 CRISPR-Cas9 system to the virus? You can put in what you think is the correct option. Okay, we have answers coming in. Chandra, Shijuni, Devadrika, Rupisha, and Oropi, all of them say that it is B, Cas 1 and 2. So let us look at it. So the general mechanism for defense against bacteria using the CRISPR system has three different steps: adaptation, expression and maturation, and interference. So what do the steps look like? As I said, post viral attack and the viral DNA essentially gets into the bacteria, and this viral DNA is the source of all problems. It will start creating more and more viruses. So Cas1 or 2 complex goes and finds the protospacer region, which it cleaves off and attaches to the CRISPR array. So in adaptation stage, the Cas1-2 complex acquires a protospacer from the invading viral DNA and integrates it as a spacer into the CRISPR array. So this is the first step. So adaptation stage, Cas1 and 2 complex acquire the protospacer. So in interference stage, the blank complex identifies the invading target DNA through complementary base pairing. Cas1 and 2, CRISPR RNA, Cas RNP, Cas9 and DNA, and spacer and Fab. So in adaptation, Cas1 and 2 play a role. In case of interference, which of the proteins do play a role? You can put in what you think is the correct option in the chat. Okay, so let us look at it. So once the Cas1 and 2 have integrated the protospacer in the CRISPR array, the next step starts, which is adaptation and the interference phase. So the interference phase, the CRISPR RNA. So what happens in between is that when the entire CRISPR array gets transcribed and they form these loop-like structures. So this loop-like hairpin loops are formed by the spacers, and the protospacers are what will be used to detect the viral DNA. They are cleaved off and then incorporated into the Cas9 complex, which is the Cas RNP or the RNA protein complex, and you can see that the protospacer from the viral DNA is present here, and it will go and try and bind to the whenever the second viral infection takes place, and whenever the second virus, so Cas9 will go and try and match this protospacer to every DNA that there is. Once there is a match, it will start cleaving of the DNA. So once it cleaves of the DNA, the viral DNA is essentially neutralized, and our CRISPR-Cas9 system has won. So this is what is the interference phase. So in the interference phase, the CRISPR RNA and the Cas RNP complex identifies the invading target DNA through complementary base pairing. Cleaving the target sequence leads to destruction of the invading viral genome, providing protection against the viral infection to bacteria. So in the interference phase, the CRISPR RNA Cas RNP complex identifies the invading target DNA through complementary base pairing. And the last question for today is: the Cas12a gene codes for an endonuclease that uses a blank to find and cleave viral DNA. The options are DNA template, spacer sequence, protein complex, or guide RNA. You can put in what you think is the correct option in the chat. Okay, so Rupsha Chandan Devatita Koel and Auditi also it is D, guide RNA. Let us look at it. So now we are going to discuss the actual CRISPR-Cas9 system. So till now we were looking at the CRISPR bacterial defense system. Now we are going to talk about the CRISPR-Cas9 genomic editing system that actually won the Nobel Prize and the one that we are majorly interested in. So the CRISPR-Cas9 system is a type 2A system in which there is this protein Cas9, which is a 116 kDa DNA endonuclease and is the sole protein required for interference. It works in tandem with a single guide RNA or sgRNA, a fusion of the CRISPR RNA and the tracer RNA. So, for the major breakthrough that occurred is that Jennifer Doudna and Emmanuel Carpentier they found Cas9, which was one of the only Cas proteins that was could do the endonuclease activity as well as the recognition activity by itself. And the second thing they did is that they figured out that the Cas systems always require two RNAs to function. One is called a tracer RNA and one is called the CRISPR RNA. The tracer RNA is essentially a hairpin loop, and the CRISPR RNA is the one that helps in recognition of the target sequence. Without the two of them, the CRISPR-Cas9 system does not work. The interesting thing that they did is that they figured out a way to join the CRISPR and the tracer RNA into one single guide RNA. So where when the tracer RNA was required to activate the system, the CRISPR RNA was required to detect the target, they fuse them together into one guide RNA. So the entire system becomes extremely simple. You just need one protein and one guide RNA, and both of them together can look for the DNA of interest and then cut it up. So it works in tandem with a single guide RNA, a fusion of the CRISPR RNA and the tracer RNA, along with Cas1, Cas2, and further five other different proteins for normal function. In 2012, Doudna and Carpentier simplified the system into a two-component system using a single guide RNA. This artificial Cas9 system, programmable for any DNA sequence, revolutionized genome editing. Further research demonstrated the reprogramming capability of Cas9 in the S thermophilus CRISPR system, enhancing its targeted genome editing. So this is the normal CRISPR-Cas9 system, and then there is the CRISPR 12 Cas12a system. So this is another new system that is currently being characterized. So what are the differences between the two? The first is the Cas9 protein is bigger in size, 4.1 kb, whereas Cas12a or CPF1 is 3.8 kb. Cas9 is a bit more complex than Cas12a. Cas9 requires both the CRISPR RNA and the tracer RNA, but Cas12a only requires the CRISPR RNA, which is even smaller, 42 nucleotides. Cas9 has two nuclease domains. Cas12a has one nuclease domain that can cut both the double strands. There is a G-rich PAM, so a NGG sequence that Cas9 recognizes, whereas Cas12a recognizes a T-rich PAM. The most interesting difference between the two of them is that Cas9 produces blunt ends, whereas Cas12a produces sticky ends. Why is this important? So, if you wanted to do a genome integration, so you wanted to not only cut the genome but cause a homology mediated addition of a particular gene inside the genome, then you put in a homology template inside along with the CRISPR-Cas system, and the bacteria or the system uses that to insert the gene. But that is very difficult again with blunt-ended products, which is formed by Cas9. But in case of Cas12a, they produce a 4 to 5 nucleotide overhang, which enhances genetic insertion and specificity of genomic repair. And finally, Cas9 cuts DNA closer to the PAM site, and this can generate indels and destroy the PAM site and prevents further rounds of cutting, whereas 12A cuts DNA further away from the PAM site, and thus there is no disruption of the PAM site, and the PAM site can be used to cut again and again the DNA. So Cas12a encodes for endonuclease that uses a guide RNA to find and cleave the viral DNA. Okay, does anyone have any questions? Okay, so that is all I had for today. So you can find the recorded video lecture on YouTube on my YouTube channel with my name in the molecular biology playlist, and I will see you the next day. Thank you for joining, everyone. Thank you.